Bismillah ar-Rahman ar-Rahim. Welcome back, friends. So we talked about pathogen recognition systems. Okay, today in this video lecture we are going to talk about the toad-like receptor in a detail. So let's start. So let me write. Now, what are toad-like receptors? Toad-like receptors are unique receptors that are usually activated against the bacteria, virus, or fungi, or any kind of the microorganisms. Whenever on the surface of the microorganism, if there are the specific molecules, we can say lipopolysaccharides in gram-negative bacteria, peptidoglycan in gram-positive bacteria, zymosin on fungi cell. We have double-stranded RNA, single-stranded RNA on the viruses. When such molecules are present on the microorganism surface, so then each molecules or each receptor or each things are detected by toad-like receptors. Okay, so usually we have almost 10 toad-like receptor. How many? We have almost 10 toad-like receptor inside the human, inside the phagocytes, we can also say. So inside the phagocytes of the human cells there are 10 toad-like receptors. So what are the function of each toad-like receptor and how the toad-like receptors are present on the cell of phagocytes? So let me explain with the help of diagram. So let's suppose this is the cell membrane of we have dendritic cell macrophages. Let's suppose these are the cell membrane of any phagocytes. Then on the surface of the cell membrane, although there are different receptors and many receptors, but here I will just draw the toad-like receptors. So there are different kinds of. First of all, there are two receptors which make a dimer connected to each other, and they make a dimer. We call that toad-like receptor 1 and toad-like receptor 2. So let me draw. Okay, now what are the function of each toad-like receptor? What actually they detect? This toad-like receptor usually detect the peptidoglycan. They are usually detect what? Peptido. They also detect triacyl lipoproteins. Triacyl lipoprotein. Moreover, they also detect zymosin. So let me write. Seen. And they also detect bacterial lipopeptides, and they also detect lipothioic acid. They also detect lipoproteins. Okay, so these are the surface molecules that are present. These zymosins are usually present on the fungi cell. So they usually detect these kinds of the surface molecules, toad-like receptor 1 and 2. Okay. Now then we have another toad-like receptor which also make a dimer, and these toad-like receptor 2 and 6 are also what? These are also the dimers. They usually detect the same things as I draw here. This toad-like receptor 2 usually detect the peptidoglycan, but they are actually present in a dimer form. Okay, except these molecules, this toad-like receptor 2 and 6, which are dimer, they also detect diacyl lipoprotein. So let me write. Okay, so after that we have some other toad-like receptor which we call that toad-like receptor 4. So let me draw here with a black marker. Sometimes this toad-like receptor 4 are present alone, but sometimes they make a dimer with their self. Okay, so that's why I draw 2. So they usually detect the lipopolysaccharide. Lipopolysaccharide in gram-negative bacteria. All these are what? All these are specially present inside the gram-positive bacteria. Then we have another toll-like receptor. Let me draw with the blue marker. So let's say this is the toll-like receptor 5. Now this toll-like receptor 5 usually detect the flagella. But keep one thing in your mind, they will detect the flagella of bacterial cell. Flagella is also present inside our own cells, means in the sperm cell there are also flagella present, or there are also the flagella present in some other cells inside our own body. So why our own cells are not detected, but the bacterial cell flagella are detected? Remember, bacterial cell flagella are made up of flagellin, and our own cells flagella are made up of tubulins. Due to this way, our own cell are not detected. This toll-like receptor 5 usually detect the flagellin proteins which are present on the flagella of the bacterial cell, inside the flagella of bacterial cell. Okay. Then we have, toll-like receptor 3, we have toll-like receptor 7, we have toll-like receptor 8 and 9. Okay, and 10 as well. The toll-like receptor 10 doesn't have any function. That doesn't have any kind of feature, but still they just detect some other protein and some other molecule. I am not explaining these other protein molecule because it is very complicated. So leave this toll-like receptor 10 because it is not understandable. Okay, then we have some other toll-like receptor which we call that toll-like receptor 3, 7, 8 and 9. Now what are the function of each receptor? So let me explain. So let's say there are the genomes of this is the single-stranded RNA. Then we have another genome which we have double-stranded RNA, and we have another genome which we usually call that CpG DNA, or we call that unmethylated CpG DNA, means cytosine guanine dinucleotide DNA. Now what is this DNA? So it is also take 1 hour to explain because it is also a separate lecture. I will explain this lecture in some other video. Okay, now just here you consider that it is the DNA. Okay, now if each of the DNA, let's suppose it is engulfed. So once it is engulfed, then inside then they make endosomes. So inside the endosomes there are the toll-like receptor 3, 7, 8 and 9. So let me draw. But remember one thing, toll-like receptor 1, 2, 6, 4 and 5, these are usually present on the cell surface outside. On the extracellular fluid they are present. But toll-like receptor 3, 7, 8 and 9, each receptor are usually present inside this endosomes. If I draw here, so how it will be look like? So it will be look like in this form. Let's say this is the toll-like receptor 3. Then we have another. This is toll-like receptor 7, and we have another. This is, let's suppose, toll-like receptor 8, and we have toll-like receptor. This toll-like receptor 3 usually detect double-stranded. They usually detect double-stranded RNA. Okay, this toll-like receptor, this is 7 and this is 8. This 7 and 8 detect what? They will detect single-stranded RNA. Single-stranded RNA. And this is toll-like receptor 9. Toll-like receptor 9 usually detect what? They usually detect this unmethylated CpG DNA. Now once let's suppose each of the things are any one of the receptors or any one of the molecule of the microorganisms are detected. Once it is detected, then what actually happen? Each receptors, each different receptors will activate, all these receptors will activate a kinds of cascader. They will activate an event of cascader, and each they will first give a signal, down signal. Each down signal will initiate a cascade event, and each cascade event will further initiate a transcriptional factor, different transcriptional factor. Okay, we have AP1, we have AP1, we have interferon regulatory factors, and we have another transcriptional factors, NFKB factors. All these are what? All these are the transcriptional factors. So once the transcriptional factors are activated, then although each cells have also the nucleus, so let me draw the nucleus here. And inside the nucleus they contain the genome, or we can say the DNA. So let's say this is the DNA of phagocytes. So what actually happen? Once AP1 factor is activated, they will sit here and they will transcribe this DNA and produce a messenger RNA. So once a messenger RNA is produced, so then this messenger RNA will translate it and they will produce what? They will produce a protein which we call that protein signaling molecules. Each protein signaling molecule will activate some other cells. So they will translate it into RNA, this one. And after that, this RNA will produce protein signal for cell signaling. Okay, after that, this interferon regulatory factors, they will sit here on the DNA of the phagocytes and transcribe to produce messenger RNA. And after that, this messenger RNA, let's suppose they will transcribe to produce messenger RNA, and after this messenger RNA, what is it is produced, then it is transferred to the cytoplasm, and after that this RNA will produce what? This messenger RNA will produce interferon, which are the chemicals, which are the proteins. We can say especially these are the proteins, or sometimes these are the chemicals. These chemicals and proteins are usually involved in the immunity as well. Okay, now after that, then we have another factor, transcriptional factor, IKB inhibitors. So they will degrade IKB inhibitor. Usually they not produce what? Messenger RNA. They will once activated, so they will reach to this one inhibitor and cause the destruction and cause the degradation of this one inhibitor. Once this one inhibitor are degraded, then what actually happen? Due to this way, chemokines, cytokines, co-stimulatory molecules are activated. Okay, so different kinds of chemicals are activated, which we call it chemokines, cytokines, and co-stimulatory chemicals. Now once each molecules are secreted, then they will call upon some other cells, and after that adaptive immunities are activated. In the previous video lecture in pathogen recognition system, if you remember, innate immunity systems are activated, but in this tool-like receptors, they will activate usually adaptive immunosystems. So once adaptive immune system are activated, so then special T cell and B cells are activated, and after that the process are under the control. Okay, so that's how the toll-like receptor perform their functions. So that's all about the toll-like receptors. Inshallah, in the next video lecture I'm going to talk about intracellular digestion. Means once each molecules are ingested or engulfed, then what actually happen inside the phagocyte cell. Okay, we call that intracellular digestion. And after that, then we are going to talk about exocytosis. All these are actually endocytosis. Then in the some other videos lecture, then we are going to talk about exocytosis. Okay, so please stay tuned and continue to watch the video. Thank you so much.